Thank you for the invitation and the organizing committee to present some of these talks. I'm going to be speaking now on radiogenomics in breast cancer, and I am Rivka Colen, an assistant professor in diagnostic radiology at MD Anderson Cancer Center. I have no relevant financial disclosures, and I'm going to focus on radiogenomics in brain cancer, specifically glioblastoma. And as we all are well aware of, glioblastoma is the most common, most aggressive primary brain tumor in adults, and comprises of approximately 20% of all intracranial tumors, despite having the trimodal treatment of surgery, chemotherapy being temozolomide, and radiotherapy, as well as advances in clinical trials. We still have a median survival of these patients at less than 2 years, approximately 14.6 months, and long-term survivors are approximately or slightly less than 10%. And this low survival remains despite genomic discoveries provided by the Human Genome Project and the Cancer Genome Atlas, also called the TCGA. And no effective therapeutic has been developed based on these genomic discoveries yet, although we do have a greater insight into gliomogenesis, treatment response, and patient prognosis. So for the patient, really little progress has been made, and the clinical course and everyday life of a cancer patient remains largely unchanged. So if we look at one of the first papers coming out looking at glioblastoma and how we can molecular subclassify glioblastoma, and this was published in 2010, we see that there are 4 molecular subclassifications or subtypes. So this was the first paper, or one of the first papers, demonstrating that glioblastoma, all glioblastomas are not created equal, and actually can be divided into 4 molecular subtypes. And these are based on aberrations and gene expression abnormalities such as EGFR seen in the classical subtype, NF1 seen predominantly in the mesenchymal subtype, which has the worst prognosis and is the most aggressive, and PDGFRA and IDH1 mutations, which are seen in the proneural subtype. And the proneural subtype actually has the best prognosis. Other common mutations and altered pathways have been elucidated based on these discoveries by the TCGA and the Human Genome Project. And we now look at primary versus secondary glioblastoma in a different way. While previously we defined primary glioblastoma as those patients who had glioblastoma with no progressive or previous lower grade glioma, such as low grade or anaplastic glioma, and these were seen in older patients. Today, we define primary glioblastoma based on their molecular composition. In this case, IDH1 wild type. In other words, they do not harbor the IDH1 mutation. On the other hand, secondary glioblastoma, previously defined as those seen in patients with a younger age and arising from lower grade gliomas such as low grade glioma or anaplastic glioma, we now define these based again on their molecular composition. In this case, secondary glioblastomas harbor the IDH1 mutation. So this is something with regards to how genomic discoveries have changed the way we define primary versus secondary glioblastoma and bring about subclassifications in GBM that are novel. Other common mutations, the 1p19q, which I'll go into a little bit in more detail, the GSIMP, and the MGMT. So 1p19q, when it is co-deleted, it is seen in 70% of oligodendrogliomas and a small subset of glioblastoma patients who have an oligo component. And these patients have a better prognosis and have better chemosensitivity to therapy. The IDH1 mutation, also called the isocitrate dehydrogenase 1 mutation, seen in 70% of grade 2 and grade 3 astrocytomas, and approximately 5% of glioblastomas, the glioblastomas being secondary glioblastomas. These also, the IDH1 mutation patients, have a better survival, and the median survival being 31 versus 15 months in those patients with IDH1 mutation versus those without IDH1 mutation. one of the interesting things now is we have MRI spectroscopy that can measure levels of 2-HG, which is a byproduct seen in those patients with the IDH1 mutation. And so we are able to identify patients with IDH1 mutation based on MRI, non-invasively being the MR spectroscopy. Glioma, CpG, island methylator phenotype, also called GSIMP, seen in around 7% of patients and seen and overlaps with the proneural subtype glioblastomas. They also are associated with IDH1 and TP53 mutations and have a better outcome. As a matter of fact, if you look at patients with the proneural subtype, they can be further divided into those patients with the GSIMP phenotype versus those without. And those with the GSIMP phenotype have a better prognosis than those in the proneural group that do not harbor the GSIMP phenotype. So this is something very interesting. MGMT, this is an epigenetic phenomenon that occurs where you can have methylation of the promoter at its promoter site. MGMT is a DNA repair enzyme and is methylated in approximately 50% of glioblastoma patients. Those patients with MGMT have a better response to alkylating agents such as temozolomide, which we currently use today as the standard of care, and also have increased risk and increased incidence of pseudoprogression. And again, this portends a better prognosis and response for the patient. So MGMT, another genomic biomarker, predictive and prognostic. But if we look at what we've done over the past decades, we do have whole genome DNA, RNA, and epigenome-based microarray analysis that at some point for investigators and scientists, it heralded an age of new and effective targetable therapeutic strategies. However, although this has allowed for greater insight into the understanding of gliomogenesis, understanding how glioma is formed and is developed, treatment response, and patient prognosis, the search for an effective therapeutic for GBM continues. So we do have what we call the genomics revolution, and how can we help as radiologists and as imagers to move this forward? So there is this relatively new field within the imaging field called radiogenomics, also known as imaging genomics. And what this does is it links specific imaging traits that we call phenotypes or radiophenotypes with gene expression or gene genomic events or genomic pathways or associated genomic networks. So this is a new, relatively new phenomenon where we can do this in a bidirectional approach and look at the different types of phenome to genome and genome to phenome correlations. So the type of correlations that can be done, we look at an imaging characteristic or imaging characteristics and link them and correlate them with either mRNA, microRNA, DNA such as copy number and mutations, known genetic subtypes or subclassifications, and epigenetic events such as promoter methylation of MGMT and other different other different genes. So imaging genomics, to a certain extent, can address some of the limitations that we are seeing within the genomic field. And as imagers, we can play a role in how we are able to really enhance patient outcomes and find that effective therapeutic, not only for glioblastoma, but cancer in general. So if we look here, what does imaging afford? So we know that imaging reflects both the underlying biological processes and tumor microenvironment. As we can see here on the right, we have the MRI feature set and we have different types of imaging analytics. And looking at the area of non-enhancing T2 FLAIR hyperintensity, that reflects in glioblastoma a mixture of edema/infiltration or invasion. If we look at enhancement, the enhancing portion of the tumor, that reflects an active proliferative portion of the tumor. Necrosis reflecting cell death. Diffusion reflecting increased nuclear to cytoplasm ratio, as well as increase in cellular density with tumors perfusion, increases in angiogenesis. So we're able to look at look at the MRI and determine and reflect as to what is the underlying biological processes and tumor microenvironment. We've shown in multiple studies, and I'll go over a few of these, that we can take that next step and correlate it with the underlying genomic composition of the tumor. Another strength of imaging is that it is non-invasive and it is cost-effective. In other words, it is already routinely done and part of the patient workup. So some of the ways that imaging can help in what I call the genomic revolution is as a biomarker or imaging surrogate, and can look at the tumor in its entirety. So one of the major issues with genomics is that we do have a specimen that is taken from a portion of the tumor. Most of the time we don't know which portion of the tumor. And we analyze that and we perform genomic analysis, but we don't perform genomic analysis on all the heterogeneous portions of the tumor, but just on the area that the specimen was obtained. We do know that there's intratumoral as well as inter-individual heterogeneity, and this is one of the bottlenecks of the moving forward of genomics as to how can we really enhance patient outcomes and find that genomic target that will really cure or help at least cause a dent in cancer treatment. So looking at imaging as a surrogate, imaging, we can look at the underlying patient genetic heterogeneity, and how can we do that is imaging can visualize, evaluate, and analyze the entire tumor, not just a biopsied portion of the tumor. And thus it is helpful and can predict if a patient has a specific genomic target and thus a candidate for this therapy. It could be molecular targeted therapy or immunotherapy or other types of therapeutics. It can guide the biopsies to the region of maximal interest and yield for genetic and histological analysis, and thus give a better representation of what is the majority representation of the genetic profile of the patient if we're able to look at the imaging and determine where are we going to take the specimen from. It can predict whether a patient can be a responder or non-responder to therapy, and can help prognosticate patients with regards to patient outcomes and survival, and determine based on the underlying genomic composition of the tumor, or even based on the image and radiological heterogeneity, what is the likely survival and outcome of the patient. So this is something that is extremely important. And I put this here. This is actually taken from the New England Journal of Medicine, Gerliging and authors, who looked at renal cell carcinoma, and just to further cement the fact that tumor heterogeneity, whether it's in glioblastoma or in this case in renal cell carcinoma, is a very important reason for why we still have to look at the entire tumor and look at different portions of the tumor in order to get a better representation in a landscape of how the tumor, what is the makeup of the tumor, and how is the tumor and the cancer likely to respond. So thus, given that we have the ability to do the imaging genomic correlations, has emerged this field. And if we look at a little bit on the history, even though we're hearing about radiogenomics most recently as of the past few years, qualitative correlations between imaging and genes have been done, the first in 2004, and then in 2008, 2 papers came out. In 2009, it was very interesting. A radiation oncology group looked at radiation resistance in genes and coined this term radiogenomics. And thus NIH more recently in 2011 asked that the radiological and imaging field change from radiogenomics to imaging genomics to differentiate ourselves from the radiation oncologist. But still, we do use the term radiogenomics and imaging genomics in the imaging field interchangeably. The quantitative imaging genomic analysis began with the TCGA with my group in 2011. And subsequently, we began the TCGA Imaging Initiative, and now we are called a TCGA Glioma Phenotype Research Group, and whoever is interested is welcome to join, as well as the ASNR Imaging Genomic Study Group, which I co-chair with Whitney Pope from UCLA, and we look at the different types and give different types of perspectives on imaging genomics and look at and study the field with regards to the science and research of correlating the imaging with the genomic composition of the tumor. So this is showing the qualitative correlations performed imaging genomics in 2008. Pope looked at incompletely versus completely enhancing tumors and found that those patients who had completely enhancing GBMs had an increased preponderance of proangiogenic genes and edema-inducing genes, as well as VEGF. That same year, by Dean and colleagues, they looked qualitatively at patients who had GBM with a more infiltrative versus edematous phenotype and found again that there was differential expression of genes in each one of these, as well as survival differences. It wasn't until 2011, the end of 2011, that we published the first paper, which was the first comprehensive imaging genomic analysis using quantitative MRI volumetrics and large-scale gene and microRNA expression profiling. And what we did here is we correlated the non-enhancing T2 FLAIR hyperintensity portion of the tumor with the underlying genomic composition of these patients. And this is the area that we were looking at, which is the blue area, which is the area of the non-enhancing portion of the tumor reflective of edema/infiltration. And we looked at the underlying gene networks. So this is a little bit more in detail about the study. So this was what we termed the invasion study. And what we wanted to do was to determine the genomic correlates and the genomic network of an edema/invasion MRI radiophenotype and potentially uncover genomic targets for GBM therapy. So we took 78 treatment-naive GBM patients from the TCGA, and we correlated looked at over 13,000 genes and over 500 microRNAs. So how we did first the image analysis is using the 3D slicer. We performed contouring and segmentation of the different phenotypes of the tumor. So using the axial FLAIR, we performed volumetric analysis of the non-enhancing T2 FLAIR hyperintense portion, and then we registered this with the T1 post-contrast, and we obtained the volumes of the area of contrast enhancement as well as the area of central non-enhancing necrosis. And then we looked at patients and dichotomized patients with high versus low non-enhancing FLAIR hyperintensity volume. And we looked at those with high versus low volume. And what we found that periostine, the gene periostine, was seen and associated with the high FLAIR phenotype, patients with high FLAIR volume, and that microRNA 219, microRNAs regulate genes, and we found that this specific microRNA was predicted to bind and regulate periostine. And we found that those patients demonstrated here high periostine gene expression, and this is its associated network, and these were the top concordant genes across discovery and validation sets, and this is the gene periostine with its associated gene networks. We looked at periostine with regards to survival, and we found that those patients with high periostine, high FLAIR phenotype, high FLAIR volume had a lower survival, they had a worse survival than those with low periostine, low FLAIR volume. And this was both concordant in the overall and progression-free survival. Very interestingly, we also looked at periostine gene expression levels in patients with the mesenchymal, which is the worst type of subclass or submolecular subtype of GBM based on the Verhoek paper, versus patients who had the proneural subtype. And we found that those patients with high periostine were predominantly seen in the mesenchymal group and those with low periostine in the proneural group. Periostine, this was one of the first papers highlighting the fact that there were survival differences in patients as well as imaging genomic correlations. And this periostine gene had previously been described very little in the literature on pancreatic cancer as well as kidney cancer, which demonstrates that it is involved in those cancers in invasion. And this is exactly what we were seeing based on our analysis in glioblastoma as well. And in figure part D, again, we're looking at the microRNA, which showed that microRNA 219 was predicted to bind and regulate periostine. Interestingly, we found that periostine was more predictive of patient survival than the subclassification schema used in GBM today, which is the molecular subclassification by Verhoek of 2010. So we thought this is something that's extremely interesting. We found these correlations in the humans, but in order for this to move forward, we have to perform in vivo and in vitro validation. And so this is the first validation of imaging genomics. So we took patients with low flare volume, and we found in vitro that they had no to little protein and very little mRNA expression of periostine. We put these cells in the animal model, and we found that those patients with low flare, when their cells were placed in the mouse model, these tumors grew to be more encapsulated, almost like metastases. So they weren't invasive, but more encapsulated, growing like a ball. Those with high flare had high periostine protein and high mRNA, and when placed in the animal model of the mouse, found again that these, similar to what we were seeing in the humans, had high invasion in vivo. And so they grew like a typical invasive cancer. And this is what is demonstrated here. We looked at perfusion imaging genomics and correlated different types of, perfusion parameters with genomic networks. And what we found was that those patients with high relative CBV had increases in angiogenic and erythropoietin signaling pathways. Interestingly, we also found that those patients with high perfusion between 0 and 1 centimeter beyond the area of contrast enhancement, in other words, within the area of non-enhancing tumor juxtaposed to the area of contrast enhancement, had a worse survival than those who had a low perfusion or lower perfusion in that region. So this is just demonstrating the migration and hematopoiesis network, as well as the erythropoietin signaling pathways. We did the same thing looking at contrast enhancement. So we obtained volumes of contrast enhancement and dichotomized the patients between those with high versus low contrast enhancement. And we found, and this is demonstrating a heat map, where we find that there is differential expression of genes in the high tumor enhancing volume versus those patients with low tumor enhancing volume. We looked also at imaging genomic necrosis. And what we found when we performed transcription or factor analysis, we found that necrosis was not the same in males versus females. That the mechanism by which cell death was occurring was different in the two sexes. So what we found was a distinct sex-specific molecular mechanism of cell death in GBM. In males, cell death, as defined by necrosis on MRI, appear to occur via an apoptotic pathway driven by TP53, and in females via a necrotic pathway driven by MYC. And there were concordant survival data showing, again, that there was worse patients or females with high necrosis had a worse prognosis than males with high necrosis. So this was something very interesting, and it was published a few months ago in Radiology. We also looked at MGMT, imaging genomic markers. In other words, we looked at the imaging characteristics in patients with MGMT methylated versus unmethylated GBMs, and we found that based on the imaging, we can predict if the tumor was predominantly MGMT methylated versus unmethylated with an accuracy of 80%. So this is something looking at the imaging and then able to predict the underlying genetic, whether it be methylation status or mutation, that this is possible using imaging genomic correlations. So this is one way doing these correlations between the imaging and between the genetic events and genomic composition, but we still haven't fully looked at intratumoral and accounted for intratumoral heterogeneity. And this has been a challenge for biomarker development and personalized medicine. And this is just, again, the picture taken from Gerlingen's paper showing that when you biopsy different parts of the tumor, there are different genetic profiles in each of these locations. So it depends, your genetic profile of the patient will depend on how many biopsy sites or where you biopsy the tumor. So tumor heterogeneity is a challenge, and we are also able to look at it on MRI. So not only using our biopsies that we're able to see the genetic differentials, but also on imaging. So this is a patient with a left parietal lobe tumor, which demonstrates areas of differential perfusion. And if we take biopsies, as demonstrated by these stars that I'm placing here, of the different regions, we get also different types of histological, cellular, and genomic profiles. So it's very important to take into account where the tumor was biopsied and which portion of the tumor are you seeing. And this is a study that we hope to be doing subsequently. When you talk about tumor heterogeneity and the ability to look at different portions of the tumor by small biopsies, you have to bring into account, so how can imaging be able to really quantify or really reflect those small microscale areas of where you're biopsying? And so thus comes the emergence of this new field called radiomics, which is a high throughput extraction and analysis of quantitative imaging features. So basically what we do here is we perform image segmentation, and the blue is the area of edema/invasion, then we have the yellow area of contrast enhancement and internal necrosis, and the orange. We perform feature extraction using our trademark radiome sequencing, and we're able to see the different niches on imaging within a homogeneous portion of the tumor. So areas that look quite similar within the edematous/invasion portion of the tumor, and areas that look quite heterogeneous, quite homogeneously enhancing, are actually internally heterogeneous when you look through the microscope or microscale analysis of radiomics. And then we're able, using our radiomic analysis, to really obtain thousands and thousands of data points, because the way we do this feature extraction is a very high-level multidimensional data extraction on a voxel by voxel or even pixel by pixel approach. So we're looking at the different pixels, the different voxels, and extracting each of these and coming up with an image of whether it be heterogeneity, homogeneity, or different other parameters. One of the important things about looking at the heterogeneity of the tumor is that we know that the niches within the cancer radiome also harbor distinct genomic profiles, and that these niches can be correlated to treatment resistance, treatment response or non-response, and distinct molecular compositions. So this novel method for automated feature extraction in GBM and in any cancers will be important going forward to be able to match the tumor and the area of biopsy with the imaging and what we're finding on the radiomic analysis. This is just very briefly showing that our MD Anderson immunotherapy cohort, we were able to use radiomic analysis to differentiate patients with pseudoprogression versus true progression at the point when there was a question of whether the patient had a recurrent tumor or was a host response. Also demonstrating the strength of radiomic analysis is given that it is a pixel by pixel or voxel by voxel approach. We are able to see things beyond the resolution of the naked human eye. So here we have a right frontal lobe tumor which demonstrates quite rounded borders, but when you perform the radiomic analysis, there is actually extension of tumor beyond the area of non-enhancing T2 flare hyperintensity. So this is, again, a strength that we can use with radiomics. Radiome sequencing, it's similar to genomic sequencing in that we assess and describe the entirety of the cancer, in this case the entirety of the imaging parameters of a given lesion, and that we term radiome sequencing. And one of the things that we can do, which I alluded to before, is be able to look at the heterogeneity of the tumor, the portions that were biopsied, and match it with the precise location where the biopsies were obtained and correlating it with the image itself. So we're able to a certain extent now decode the imaging landscape in glioblastoma and in cancer in general. And this is just looking at image-guided, image-registered stereotactic biopsies, which demonstrates points of where the biopsies were taken and their matched imaging characteristics. One of the things in the American Society of Neuroradiology, or the ASNR, that we're doing in the Imaging Genomics Study Group is forming an imaging genomic consortium where we will begin a stereotactic biopsy project. And this would include 10 centers, and many people have been committed and been involved in this stereotactic biopsy project. And so this is an extremely important endeavor. And if anybody is interested, you should contact me, and we'll be happy to have you be a part of this. So one of the ways that we can look at what is the novel imaging and imaging genomic and radiomic paradigm where we can really help enhance patient outcomes is a patient comes in, we obtain an MRI, we have our raw or DICOM data, we perform image segmentation, perform our texture analysis and radiome sequencing and feature extraction, analyze the data, and integrate the data with other similar type omics data, whether it's proteomics, genomics, metabolomics, and clinical information, all to then enhance the clinical decision making at the point of care. In other words, be able to determine should the patient be receiving this therapeutic or should the patient be changed, what is the likely outcome of the patient. In conclusions, imaging genomics can help advance the genomics field and can be correlated with the underlying genomic composition of the tumor. And given that we have validated imaging genomics in the preclinical animal models, we can use this for target discovery tool and use MRI as an imaging surrogate for patient care and possibly clinical trials. We know that not only known correlations, but new pathways and genomic targets can be uncovered and subsequently be submitted to industry for drug development and pathway discovery. Creation of a more sophisticated and precise imaging analysis, such as texture and heterogeneity analytics, can be anticipated to allow for more robust and precise genomic correlations and uncoverings of the tumor heterogeneity itself. Future directions is to continue performing these imaging genomic analysis. Always remember we have to perform in vitro and in vivo validation and prospectively do this in patients using targeted image-guided biopsies, all to create and hopefully be able to cause a significant dent in cancer therapy and enhance and increase patient outcomes and enhance personalized and precision-based medicine. One of the things when we talk about precision-based medicine and precision-based, specifically imaging genomics, is we have a lot of data. And big data analytics and pipelines for this is important to develop, and this is something that we're working on currently. So, and as we go forward, hopefully we will be able to really enhance patient outcomes. So I would like to thank my lab members and everyone in the audience. Thank you very much.